Kyprianou. That's from Mount Sinai School of Medicine in New York. She's this year's Principal's Visiting Scholar. So she's a distinguished and highly decorated scientist and also an award-winning educator and mentor. With a 50-page CV and 270 publications, I think that it would take an entire hour to go through everything she's done. But I'll just a couple of highlights. She's had a number of highly responsible roles, including vice chair for research department of urology, which she does now, as well as editing many journals. She's been the president of the Society for Basic Urologic Research and the chair of the U.S. Department of Defense Integration Panel, which decides actually where to give the money after the reviewers are done with their business. That must be politically and scientifically challenging. And another title is Colonel, which I don't really understand, but I thought that was really unusual. She's known for groundbreaking discoveries in several areas, including TGF beta signaling and androgen receptor signaling, EMT as well, and the development of treatment resistant prostate cancer. And having had the privilege of spending some time with her yesterday and today, I now hypothesize that the foundation of all of this achievement stems from her profound enthusiasm for science, which I invite you to sit down and enjoy.
Well, thank you very much, David, for this wonderful, really lovely and genuine introduction. It's an honor and a delight to be here. It's my first time in Kingston, and it's such a beautiful university town, and the weather is gorgeous, and the autumn leaves. So just thank you very much for having me, and thank you for attending. And so today we're going to talk about, no, there is not a phone, but this is, some of you who met with me today know that I have a very strong voice, so I can try and yell if you don't hear me well, but we're going to share with you some of the recent studies. Actually, some of the studies have been going on for the last decade or so, but some more recent, also more unpublished data on the intersection between epithelial mesenchymal transition or phenotypic changes, phenotypic configuration, and cell death that really can be exploited, these interactions, to overcome therapeutic resistance in prostate cancer. The focus of my research has been for several years now, in addition to some of the signaling pathways and exploiting apoptosis mechanisms to overcome therapeutic resistance in advanced prostate cancer. This is the Annenberg building where my lab is on the 24th floor, and the floor on the top is the 25th floor where the dean's office is. And you have great views to Central Park and to George Washington Bridge. So, but I know you have beautiful views here. So, and maybe some of you have seen this in terms of the hallmarks of cancer. This is the most recent update that came out last year by Hanahan. In the first version, it was only about 10 processes or hallmarks of cancer. Now there are multiple. But what I would like to focus on is, like, and what has been the focus of my work for several years, is really understanding or the phenotypic changes, phenotypic configuration of advanced cancer, prostate cancer, and also the mechanisms resisting cell death, because resistance to cell death is equivalent or is translated into resistance to therapeutic options that we can offer to patients with advanced prostate cancer. So what is the current treatment landscape of castration-resistant prostate cancer, the most lethal form of the disease? And what do we know so far about what the therapeutics are available and the impact they have on patient survival? Because this is what we do, translating things from the lab to the clinic. So as some of you may know, with Dr. Berman here, is that what kills men with prostate cancer is metastatic disease to the lymph nodes and the bone, okay? And these are the clones that become ultimately resistant to the current therapeutic approaches, besides androgen deprivation therapy. as well as docetaxel, taxane chemotherapy, and we'll talk about this in a second. But unfortunately, and here is what's clinically available, the current treatment therapy, the current treatment approaches, including docetaxel, immunotherapy. I know we'll talk with some of you about immunotherapy, anti-androgens such as enzalutamide and abiraterone, as well more recently, the different radium-223, apalutamide, and enzalutamide and darolutamide that are versions of enzalutamide, which is a first generation anti-androgen. And more recently, the FDA has approved the PARP inhibitors. So if you look about not only the cost of the patients, but in terms of increasing survival in patients with advanced prostate cancer, is really minimal. It's only a few months. And these patients are in a lot of pain. So clearly, with all that is invested in terms of research and big pharma efforts and billions of dollars, we need to do better than that. So this is the message I'm here to convey in terms of what we do with translating the things we're finding in the lab to the clinical setting. So just a brief history of how the treatment modalities for prostate cancer over the years, since the 90s, when docetaxel and cabazitaxel first came out as the first and second line taxane chemotherapy. And then more recently, in 2000, a few years later, we have Gleevec as the Intersecting. Of course, these were available for the treatment of breast cancer. And in 2014, where we're turning into immunotherapy. All these, of course, there is a lot of excitement about all this sexy appearance of these different drugs that can really cure cancer. And most of them can also impact prostate cancer, gracing the Time magazine, the cover. So where we are today in 2023, is these are the therapeutic options we offer to patients with metastatic castration-resistant prostate cancer. And this is what I will try to do is how can we exploit some of the mechanisms that are really activated in response to these agents, maybe in combination strategies that by causing these phenotypic changes at the cellular level can have a therapeutic impact and increase survival in patients with advanced disease. So what we have again is docetaxel, the first line therapy, taxane chemotherapy, and then we have the treatment given this is after they fail anti-androgen treatment, and we have the enzalutamide, what the abiraterone, these are first generation anti-androgens, and radium and PARP. So all you do is to really give these patients with advanced disease, you keep hitting them with all these treatment modalities. Now, after failing this, then we give the second line chemotherapy, that is cabazitaxel, and maybe either alone or in combination with some of the other anti-androgens, as well as radium more recently. And now there is some question about this PARP inhibitor, whether they have any. It's been approved by the FDA for treatment of having this, of course, like PARP inhibitors having high microsatellite and instability. And also Pembro, which is immunotherapy. I know some of you work on immunotherapy. There is a lot of interest. I don't work in immunotherapy. My chairman, Dr. Tewari, does. And there is a lot of interest. But we're not here to talk about immunotherapy. We're here to talk about how maybe a combination of these different therapeutic modalities, given at the right time and the right sequencing, can really have an impact. So the challenges we all face, and I know you know from David's work as well as others about prostate cancer, is the phenotypic plasticity and the cell heterogeneity that characterizes as a solid tumor that is a very different dynamic from what we see in other solid tumors. So many years ago, one of the very talented medical MD PhD students who worked with me, a pathologist, and now she's attending at Memorial Sloan Kettering, Dr. Meng Zhu, in collaboration with Tom Beer at the time when he did the first clinical trial, while he was in Portland, Oregon, about the treatment, giving patients with advanced prostate cancer and then undergoing biopsies, given the first-line chemotherapy, docetaxel, provided the first evidence that docetaxel, and I'm sure that some of you may know, as a taxane chemotherapy is known to be attacking the microtubules. So what Mengli was able to show, both in the clinical specimens as well as in vitro models, that this chemotherapy, taxane chemotherapy, is also able to affect the androgen receptor distribution, the cellular distribution. You can see here in clinical specimens, nuclear staining is present with high intensity in the treated controls. However, after 4 weeks of treatment with docetaxel, there is quite a wide distribution of the androgen receptor signaling in the cytosol. And this is like the quantitative analysis of the data. So this was provided the very first evidence that docetaxel is a first-line taxane chemotherapy, in addition to causing the microtubule instability that is known to be affecting the treatment response, it can also affect the androgen receptor activity and distribution. How does it is happening? And again, I'm summarizing this. It has been published, but it sets the foundation of what we know about the current drugs and how we can combine them. So in terms of the way it interacts, the way that the AR is targeted by this docetaxel treatment is because it's able to bind the tubulin, which is a major component of the microtubules, via the N-terminal domain of the androgen receptor. And here is a structure of the AR, and I'm sure some of you work on that molecule, but we've shown that it's the N-terminal domain of the androgen receptor. All the other targets of the AR, the anti-androgens, are known to bind or interfere with the ligand binding domain of the androgen receptor, where it binds to the DHT, which is the active androgen. So with that in mind, we started a collaboration with another famous Canadian, Dr. Marianne Sutter in Vancouver. At the time, she was developing this drug, what is called the EP001, that it was a specific inhibitor that is targeting the N-terminal domain of the androgen receptor. And this is Dr. Sarah Martin, who was a PhD student in my lab at the University of Kentucky. And what we can see is that, again, using the xenograft models of advanced castration-resistant prostate cancer, the combination of this EP001, the anti-androgen, that is in combination with taxane, docetaxel, is causing a severe suppression of the xenograft growth that is really more significant than either drug alone. And the way it does that, of course, is causing apoptosis. But prior to that, there is this very dynamic interconversion between causing EMT, the epithelial mesenchymal transition, back to mesenchymal epithelial transition. And when you give the combination, this is what you're observing. And again, a lot of you, we talked about with some of you today about cell plasticity. EMT is becoming a very interesting process right now, but we all have to remember that this is a very transient phenomenon. So this was published a few years ago, but the whole idea is that we have this combination treatment that is really attacking the cells at the time when they undergo this epithelial transition, making them more vulnerable to the subsequent treatment with taxanes, and I will explain that. So what Sarah did later, we tried to use whether cabazitaxel monotherapy can actually have, and these cells are known, the 22RV cells, these came from castration-resistant prostate cancer, and they're known to be resistant to taxane, to cabazitaxel chemotherapy. That's why we tried the combination. So where is this coming from? So you can appreciate here, this is a quantitative analysis. You can see this is that is cabazitaxel chemotherapy is actually inducing this is the proliferation. It's actually promoting proliferation, and that's why the cells are resistant on these xenografts. Now, when you give it in combination with enzalutamide and cabazitaxel, there is a reduction of proliferation, and at the same time, there is induction of apoptosis. So again, it's very hard to, when we talked about this today, about really tracing this phenomenon of not only apoptosis, but the epithelial to mesenchymal transition. And what we see is that cabazitaxel, in a very similar fashion, when given alone, like the docetaxel, is causing, is retaining some of the nuclear AR localization. again, these are castration-resistant prostate cancer cells, but they do harbor not only the AR variants, but also they harbor the good amount of the regular androgen receptor. And what is this? Cabazitaxel is also promoting, if you will, the induction of ecadherin, which is a marker of proliferation. So that means the cells are going back to mesenchymal epithelial transition, and when given the combination treatment, then one is able to target that, and therefore having some affordable, some accountable activity. Now, so what I'm coming to that is that the sequencing of cabazitaxel with androgen deprivation therapy and anti-androgens can impact the EMT and promoting this phenomenon of plasticity in therapeutically resistant prostate cancer, enabling them to achieve some kind of therapeutic vulnerability. In collaboration with, collaborations are always good, getting PDX models from MD Anderson with Dr. Nora Navon, who is an expert and she published widely in that. So we got a series of PDX models from her database, and these are all from patients with lethal advanced prostate cancer. And some of them received the treatment that you can see up there. Again, they all have very aggressive disease. Some of them in sequence, they receive androgen deprivation therapy and then cabazitaxel, after docetaxel failed. And what we see is that in the treatment naive, what we have indeed the mesenchymal phenotype, the epithelial phenotype, mesenchymal to epithelial transition predominates, as you can see here from the presence of ecadherin and genetic. And all the other lethal tumors, you can see that there is high EMT activity. However, if you look at this, where metastatic castration-resistant prostate cancer, these are PDX models, when they're given the cabazitaxel, post-abiraterone, and post-androgen deprivation therapy, this is really, so goes back to the timing, there is this manifestation, if you will, of the mesenchymal epithelial phenotype, the epithelial phenotype, that then can be subsequently targeted by other treatments. So this is what we're finding in our xenograft model of castration-resistant prostate cancer. It seems to be the same for the PDX models, that these xenografts, the epithelial phenotype has to predominate upon hitting them or induction by cabazitaxel, and then it can be subsequently treated with another anti-androgen or another therapeutic approaches I will show a little bit later. So at the same time, when these preclinical data were coming out, a very nice study by Chris Sweeney, who is one of the best medical oncologists in prostate cancer field. Now he moved back, he was at Harvard, now he moved back to Australia, he's native. So it shows that the response to docetaxel and the androgen deprivation therapy, it happens, it can be more effective, if you will, more pronounced if it's given to chemotherapy naive patients. This is hormone-sensitive prostate cancer. So it goes back to when we're dealing with hormone naive, before they were exposed to any ADT, before they are exposed to anything, when you hit them with cabazitaxel or in this case, docetaxel, the taxane, you can actually have a significant increase in survival. This is for high volume disease, but you can see that there is increase in survival up to 17 months. If you remember for one of the early slides, we were dealing with increases, with just given each drug given alone, only about 3 to 4 months. So this clinical trial received a lot of notoriety, a lot of attention, because it was the first evidence that you can really have a substantial increase in the survival of these prostate cancer patients. So timing and sequencing of treatment is everything. So, make sure that. So, what are the lessons we learned from clinical trials? I know I saw today that there is a very big clinical trial center here at your institution. So I was delighted to see that. We also have a clinical trial center at the Mount Sinai. But what are the lessons we learned so far in terms of overcoming this resistance in lethal prostate cancer? So maybe the problem has been because so far we've been focusing in targeting the castration-resistant disease, the advanced prostate cancer, and then we see minimal effect in survival. All this, and some of you probably are familiar with the studies of attitude, the Stampede, the Titan, they have given this fancy names. But maybe it's about time that we focus about targeting with chemotherapy induction, the castration sensitive, the hormone-sensitive disease in order to see really a significant effect in survival. So going back to Sarah's models, and I know I talked to some of you today about model. This is the TRAM model that is TGF-beta driven with advanced EMT phenomenon. And this is what Sarah was able to find, that when you take this model, this preclinical TRAM model of TGF-beta driven EMT that really developed very advanced, almost neuroendocrine phenotype prostate cancer, it can be reprogrammed to undergo redifferentiation by cabazitaxel-induced mesenchymal epithelial transition. So the acquisition of met with epithelial phenotype in this advanced model then can enhance the response to ADT, to anti-androgen treatment, orchestration. And again, this goes back to the dynamic of how this, and I know most of you are familiar with, about the changes in ecadherin and ecadherin, that is really a fine balance of the phenotypic or plasticity or whatever phenotype you'd like to define. But what was very interesting is the observation that we made that in response to cabazitaxel, and because of this reversion back to the epithelial phenotype, there was also an induced redifferentiation, if you will, based on cytokeratin 18. And this is the responsibility, and this is what really drives. I remember when Sarah first made that observation, I called another good friend of the pathologist at Johns Hopkins, former colleague of David, Angelo De Marzo. I said, do you see that in response to chemotherapy? Do you see the redifferentiation of prostate tumors? And he said, well, we don't know. This is not really very well known or very clarified or well studied. And we believe that when given to this, after androgen deprivation therapy follows cabazitaxel treatment, then what you have is a significant decrease because you're targeting the epithelial phenotype now. You're not keep hammering, targeting the mesenchymal phenotype that are known to be resistant. We have this acquisition of the epithelial phenotype that can be more vulnerable, if you will, to anti-androgen treatment. And again, a couple of years later, this is from Lu Chen, a very prominent investigator from Memorial Sloan Kettering. And this paper was published from this group, indicating that this reprogramming of the prostatic cancer cell fate, as we call it, that is really promotes this luminal differentiation of prostate tumors. And this is without any androgen deprivation or without the albumin. And it's always good when your ideas or the concept can really be validated or be confirmed by other investigators. But this is the main concept here, that this reprogramming back to the epithelial phenotype can promote this luminal differentiation I showed you previously, and therefore these tumors can be really more vulnerable to subsequent treatment. So Sarah really wanted to know what is the mechanism of this, what is really driving this, and using again some of these hormone-sensitive prostate cancer cells. These are the VCAP cells. And what we can see here is that, first of all, MDB is the old name for enzalutamide. But what was interesting, of course, we know that enzalutamide in the presence of DHT, there is a high retention of the nuclear androgen receptor. When you hit them with cabazitaxel, you can see that there is some partial nuclear AR retention, but then after a longer treatment period, you can see here that it disappears. So the whole idea is that giving the cabazitaxel in the presence of DHT, yes, it holds some back of the androgen receptor, meaning that the AR activity is still there, and then you really expose the cells with the epithelial phenotype now, they have some AR around, then you treat them with androgen deprivation therapy or anti-androgen, and therefore you see this therapeutic or anti-tumor effect. The other observation that Sarah made from this is that cabazitaxel is causing this multinucleation, okay? Multinucleation, that means the mitotic spindle are involved, whatever, but it was quite fascinating to see that the drug alone is causing this, and maybe that's part of this vulnerability. So we wanted to see what is really responsible for this multinucleation, and you can see that this is really the centrosomal amplification that is driven or is navigated, if you will, by these mitotic spindle proteins called kinesins. And you can see here that in the presence of the anti-androgen as well as cabazitaxel, you do see this multinucleation, and you can see from the increase in pericentrine, which is the marker of centrosomal amplification, there is a massive centrosomal amplification when given, and then after 96 hours of treatment, which is something that is really driving the therapeutic effect, you can see that this is a little bit becoming a totally amplification. But what happened to kinesis at both at the mRNA level? You can see there is a transient increase in response to treatment in the presence of PHP, and then with longer treatment period, when the cells undergo apoptosis, there is this loss of kinesin that is also reflecting the loss of the mRNA, but also at the protein level. The two major kinesins is HZ and MCAS, you can see here, but these are involved during the separation of the mitotic spindle. And so also responsible for causing the integrity of the centrosomal amplification. Again, this data was published. The interesting kinesins that will also become more prominent later on about their role in therapeutic resistance is that a few studies, and when we did the TCGA analysis, have shown that kinesin, the first one, KFC1, is associated with poor clinical outcomes in these prostate cancer patients, poor survival. And then, so these are the proteins, this is just a cartoon to show you that these are the proteins that are helping kinesis, are helping to move along or to walk along the microtubules and delivering whatever cargo they're carrying, in this case probably AR, as I'll show you in a minute, along the microtubules. That's why they're targeted by the taxane therapy into the nucleus. So very important, make a really very important and rather underestimated role. So they play a role in the microenvironment. And I know I talked with many of you today, and you're familiar about the importance of the tissue of the tumor microenvironment in maintaining homeostasis, as defined by Claude Bernard, a famous French physiologist, the milieu intérieur. And I don't need to remind David here, this audience, about the microenvironment in prostate tumors or in the prostate gland. Of course, we have the basal cells, we have new cells, we have the glandular epithelial cells. But all these contribute to therapeutic resistance, and how can we target them in terms of really overcoming the resistance. So we turn to TCGA. I know there are a lot of bioinformatics experts. One a very talented graduate student, she just graduated a month ago, Dr. Tina Chen, who is a bioinformatics student. Her his co-supervisor with Dr. Navneet Dopra, and you can see that she did this trying to identify the prostate tumor microenvironment contribution to the therapeutic resistance, studying or looking at the TCGA data. And we found indeed, this is in prostate cancer, that kinesins such as KFC1 and KFC2, as well as this group of transporters that SLC, and there's a whole family of them, SLC, O1, B3, these are really lead the way in terms of being overexpressed in advanced prostate tumors, actually with the localized prostate tumors, but also with advanced high risk for progression compared to normal tissue. And again, this is what we get from the TCGA analysis. So the next thing really we wanted to, this was done in the middle of COVID, going back to the targeting of cabazitaxel post androgen deprivation therapy, or really looking at the manipulations here of in terms of what happens to kinesin levels in hormone-sensitive prostate cancer. And again, this is a LNCAP. These are LNCAP cells. These are hormone-sensitive, but they're derived from metastatic prostate cancer. But what I would like, this is a 22 research, and you can see, although there is an induction of apoptosis, when cabazitaxel was given after, after androgen deprivation therapy, there was not really much of an effect in cell proliferation because it's very counteracting, and really there is no effect on kinesis. If you look at the hormone sensitive, going back to the early stage of prostate cancer, the hormone sensitive are the LNCAP cells, you can appreciate there is a significant induction of apoptosis, a significant loss of cell proliferation, that really resonates with a significant reduction in the immunoprofiling or the expression of the kinesin. So this is, how does it happen? So this is the work that was done by the graduate student, Dr. Diane Beckerman now, who finished her PhD at Mount Sinai with me. And now she's back in Kentucky, working for a biotechnology company. So what Diane wanted to see is really looking at this interaction, and you can appreciate here that there is an interaction between the paclitaxel, the taxane, second-line taxane chemotherapy, is able to target kinesin specifically, as said, as well as the interaction of the androgen receptor in the presence of DHT in this hormone-sensitive prostate cancer cell. It's also affecting or is inducing the loss of Kyprianou, indicating that its ability to, it's inducing Kyprianou, so indicating that by itself, but then that indicating that there is the predominant, the epithelial phenotype predominates. But what, just the diagram here, I just want to say this is using a specific inhibitor for kinesin, not paclitaxel, but you can see that specifically it kind of brings it down, and this is looking at the interaction with the androgen receptor, and PSA, which is the ultimate, I guess, endpoint for androgen receptor activation. So what we think is happening, that paclitaxel can interfere and have evidence to support this, and we'll show you in subsequent slides, that paclitaxel is interfering, or this transporter that is interfering with the entry of the drug into the cells, and therefore causing the lack of response, if you will, to the therapeutic action of paclitaxel. Can really manipulate or impact the kinesin expression and the interaction of kinesin with the androgen receptor. And by doing so, kinesin means the HZ protein that we see here, by interfering with that interaction, but we see it is really causing the loss of expression, but the loss of expression of PSA. That means the interaction of, when interaction of HZ and the androgen receptor is targeted, then you see this inhibition of activity of the androgen receptor happening in hormone-sensitive prostate cancer cells with an active androgen receptor. So these are published data. What Diane did is really do some functional studies indicating that when you lose the transported protein, that you can see that it's also driving the receptor resistance to paclitaxel. So this is just a dose response in terms of cell viability. So you can appreciate that two of the clones have this enhanced resistance or lower loss of cell viability or enhanced lower cell death when the transporter is knocked down. These are used in the PC3 cells. When we knock down the transporter, you can see here that there is paclitaxel resistance. There is another growth of the cells, indicating that that there is, and also there is an increased invasion potential in the PC3 paclitaxel resistant cells. So it's in the paclitaxel resistant cells in manufacturing or exhibiting this loss of the transporter. And this is TCGA data indicating that the deletion of the transporter gene of the LCLC1B3 is really is associated with poor survival. And again, these are published data, but the paper from Europe in European urology was published a few years back, showed that this loss of transporter, there is a lot of interest and there is a whole family of them, but it seems that this loss of transporter drives the taxane resistance in prostate cancer. This is a group from the Netherlands. So this transporter not only takes the drug in, but also can be responsible for driving the androgens in and out. So with that, going back to our model, this work was done by a postdoctoral fellow in my lab, Madison Archer. You can see that this transporter, the LCCB13, and the kinesin, their profiles really correlate with the therapeutic vulnerability. These are the PDX models we got from Anderson, again, patients with advanced disease. And I would like to draw your attention here to the one that showed the AET, the epithelial configuration that was partially responsive. Again, given paclitaxel after viraderone and after androgen deprivation therapy, you can see there is an increased AR, the AR is present. So this is what's happening is totally independent of the androgen receptor. There is a severe decrease or significant decrease of kinesin, the HZ, and at the same time, there is some partial presence of the transporter. So in the other specimens where the transporter is gone, these are the ones that manifested the more lethal phenotype of the disease. So we believe that there is is this evidence, there is some support that these two proteins are really interacting, contributing to the development of the loss of transporter, as well as the overexpression of kinesin can really drive therapeutic resistance. So this time, about a year ago, last summer, the FDA had approved this on the basis of the Erasmus trial. It's a randomized, multi-center, double-blind, placebo-controlled clinical trial in patients with metastatic hormone-sensitive prostate cancer. What I mentioned earlier about the timing is everything. By the time the prostate cancer, the tumors become metastatic and resistant, it's too late to really try to experiment or manipulate or exploit the therapeutic combinations. So we need to hit them when the patients have hormone-sensitive disease. And then the FDA, August last year, approved darolutamide in combination with docetaxel, given in patients with metastatic hormone-sensitive prostate cancer, and who had already received ADT. So we'd like to think some of our preclinical data really led up to that. So now I'm coming to, I know I talked with some of you about the TGF-beta. We're talking about combination treatment, not only taxane with anti-androgens, but TGF-beta, as I mentioned, using this model of the Trump-driven, that is a double transgenic, that is the conditional loss of the TGF-beta receptor 2, that is driving the progression of the Trump model, of the prostate tumor model, to aggressive disease by promoting the EMT. So in collaboration with a very talented medical oncologist from Johns Hopkins, Dr. Channing Taller, we also had a clinical trial, but she really developed this and worked on this preclinical model first. And also Dr. Antonarakis, who now has people removed from Hopkins, the clinical trial. But we wanted to see how these tumors are targeted first by TGF-beta. And there is a very known inhibitor called Plasticity, and I know I talked to some of you today, that is specifically targeting TGF-beta receptor 2. So you can see these tumors are androgen, they are expressing, the vehicle expressing a lot of androgen receptor that is targeted by enzalutamide. It has very advanced, very aggressive tumors. Galucinergic doesn't do anything to androgen receptor. However, when you give the combination, you can see there is a depletion of the androgen receptor. But what galucinergic does, the TGF-beta EMT promoter or reversion of the promoter of the promotion of EMT is causing this cytokeratin 18 manifestation, which shows that it's really driving the redifferentiation. So maybe using this combination or this treatment that is causing this redifferentiation first, and then you hit them with the anti-androgen, this can really help the therapeutic effect. Again, this was published a couple of years ago. And we can see here, this is the combination treatment. Of course, the expression of that is when you give the combination with TGF-beta inhibitor, as well as anti-androgen, there is a decrease in proliferation, and there is a significant increase in apoptosis that we believe can drive the therapeutic effect. And we have a clinical trial going on at Johns Hopkins right now. This happened in the middle of COVID, so we're not able to recruit a lot of patients. I think we have a few patients so far. And at the same time, this is also, there was a study that came from Jindan Yu at Northwestern, and that Schaefer and Dr. Catalona, where they found very similar results using the TGF-beta inhibitor and this reversion of back to the redifferentiation phenotype. Using this model, because again, this is a model of very advanced disease, very advanced prostate cancer progression, we are also able to show that the TGF-beta combination with the TGF-beta inhibitor, Plasticity, and is affecting this molecule known as coupling. We've shown a few years ago that coupling is a protein that is involved in the cariopodia, which is, in Greek means little fig, that are involved in the progression and the movement, if you will, and the invasion properties of cancer cells. And what we have shown that, of course, since the target for TGF-beta, you can see that coupling is really in the combination by TGF-beta inhibitor as well as by combination. There is a significant decrease in the expression of coupling, which really can be translated into suppression of the metastatic and invasion potential. And again, this paper was published and using this model. So the bottom line, especially for the students and the residents, we can learn a lot from preclinical models before we move to the human disease. So in terms of human targeting, so in terms of just wrapping up this part of the presentation about this phenotypic reprogramming, in order to overcome prostate cancer therapeutic resistance, we believe that it's very important to target the hormone-based prostate cancer. Yes, maybe we're using an apoptosis-based response, but during tumor relapse, when we give them the same chemotherapy, and we need to focus on the ENT to NT conversion when given hormone-based prostate cancer, in order to really have the ENT, the epithelial phenotype, that is really afforded by the redifferentiation dynamic that I just showed you, can really be responsible for the proliferative expansion of these resistant clones or subtypes, if you will. And then we can target them with other more novel inhibitors such as TGF that are really targeting the EMT or the MET phenotype, reversing it back to MET. So what I know everybody's doing, and some of you are doing it here, the technology is leading the way, in terms of really defining some of these phenotypic signatures at the single cell level, in order to identify is this reprogramming going to be taking place in this lethal prostate cancer, or maybe in hormone-sensitive prostate cancer before it becomes lethal. And this is what we're currently at in collaboration with Dr. Navneet Dopra, who's an expert on single cell and bioinformatics, really defining some of these. This is taken from tumors, from prostate cancer, maybe defining some of these clusters of these EMTs, such as keratin, not only keratin, but notch pathway, as well as the cadherin. So defining the clusters that can really give us some idea about what is the phenotypic reprogramming happening in these tumors. And I would like to finally finish some of Madison's, so it's Dr. Madison Archer, who is currently working on really defining and using different models to identify the therapeutic resistance, the taxane, even taxane, and how can we overcome using in vivo and in vivo models. Now, these models is a C4-2B cells, the derivatives of the LN-CAP, and also there is a C4-2B that is taxane resistant, and we got those from Dr. Alan Gao from UC Davis. And what we see here is how cells, the parental cell line is taxane resistant. You can see there is 24 hours of treatment, this is 48 hours. In a concentration-dependent, dose-dependent fashion, which the parental, of course, they're really totally sensitive to the drug. But here there is a gradual increase in the sensitivity to the drug, the carboxyl with increasing doses. Of course, we're focusing only on the lower doses that can be clinically relevant. But what you can see, there are some dramatic changes in terms of happening to some of the key players. But I would like to draw your attention here to HZ, which is the protein and the protagonist of interest here, kinesin. But you can see that in the parental cells, within 12 hours, there is a significant decrease in response to treatment. Nothing is really happening to the carboxyl resistant cells. So it seems that this is the ultimate target. If we find ways of really targeting this kinesin, we can promote the cells into a more sensitive or vulnerable, if you will, phenotype. And of course, there is targeting in the P2, the apoptosis suppressor, but not much in the resistant cells. This is very recent data. This is coming from in vivo. You can see the Madison's work. You can see that the parental cell, in response to treatment with carboxyl after 2 weeks of treatment, there is significant suppression of tumor volume, and the resistant, the resistant or taxane resistant C4-2B cells, there is really is not a significant suppression in really validating, and we have some more in vivo experiments going on really to validate this. And also we turn to transcriptomic profiling by RNA-seq to really define the resistant phenotype in these prostate cancer cells. And again, we go back to what we found specifically. There is, the phenoblast, we see some of the kinesis, but some of the transporters being differentially expressed in the resistant cells which are treated. This after 12 hours of treatment. But you can appreciate here, going back to the transporters, that there is a significant, really, loss in the resistant cells, and this is what I showed you earlier about the PDX models, as well as our cell models, and that even before they're activated, but they're also, like the doctor is causing this massive reduction in this associated with the resistance. If you keep it while in the parental cell, there is a gradual, I guess, decrease, but not really significantly, higher than the resistance system. Some of the pathway association analysis that we are currently exploring, but again, goes back to how these H-cell proteins, having their precious cargo, in this case, it could be under the receptor, maybe they're facilitated by the transporter, to give, rise to some of the mechanisms that are associated, or some of the gene profiles that are really promoting the DNA repair. And this is double trifecta. This is the results coming from the RNA-seq analysis. And again, just want to emphasize that although there was enhanced apoptosis in the C4-2B as expected compared to the resistant cell, but there was also this enhanced DNA repair mechanism in the resistant cell. Okay, what interesting enough, what they had in common is the stemness, or the EMT phenotype, and that goes really back to timing. I know this is in vitro work, but we're dealing now, since we found that this DNA repair this pathway seems to be super activated using the PARP inhibitor in combination with bisphenetib, which is the kinesin inhibitor, as well as cabazitaxel. And what we're finding, there are really some profound changes happening in response to the trifecta, as we call it, in response to the triple or the three-drug combination, that there is loss of cadherin, goes back to the loss of differentiation. There is a significant decrease in HZ. There's also a significant decrease in the BCL2. But what is happening is that, while this is happening maybe in response to cabazitaxel alone, after long treatment in the sensitive cells, in the resistant cells, the only time we see this profile of all these key proteins being downregulated, including HZ and BCL2, is when we give the triple combination. So, yes, are we surprised that the DNA repair mechanisms are overactivated in the resistance? Not really, but since all these drugs are currently approved by the FDA for different cancers, maybe something that we should consider doing in terms of unlocking that phenotypic plasticity. And this is some very recent work done by another fellow in the lab, Dr. Kyprianou, while using different models. And the whole idea, and I just want to introduce you about the whole concept of analysis, which is cell death when the cells become detached from the extracellular matrix. But how this is the EMT process and analytics are different correlated. This is using in response to TGF-beta alone, and also in response to the 50 information that we developed in the lab, in specifically causing analytics, via activation or suppression of the SARC signaling pathway. And historically, SARC is one of the major players of analytic induction. But interestingly enough, when you give a combination, and this is just cell viability, but when you give a combination of TGF-beta, so you're inducing the EMT, and then you hit them with the analytics drug, then you do get a significant increase here in terms of cell death. And this is some of the work that was done in an in vivo model, again, with cow mouse given by TGF-beta. Again, when you give the analytic reagent, we give 50, as we call it, there is this massive, there is some induction period that is reconfiguring into the phenotypic, into the phenotype with, and the antigen receptor is not being involved. But also is called going back to the redifferentiation process, as you can see in the lower panel on the basis of cytokeratin 18. So, so we find these pathways that can really go independent of the AR, and yet there is somewhere an intersection between these two pathways, between analytics and identifying these players that can be involved. And finally, the last couple of slides I have, going back to the microenvironment, and I don't know if any of you are working on extracellular vesicles or liquid biopsies, but this is Dr. Navneet Dopra, one of the leading experts on extracellular vesicles and characterization. This work was done by a very talented medical student who worked with me and now is applying for a urology residency, Simita Deglani, and it was published last year. In terms of really profiling the microenvironment, the differential expression of the differential expression of the disease between cells in the extracellular vesicles. This is using the term uses to how we identify this is identify the extracellular vesicles by microscopy. And from microscopy, you can see these are really secreted vesicles. They're quite small, but there is a significant increase in the extracellular vesicle secretion in the tumor versus the benign prostate. And this is a 2012 result, a very advanced castration-resistant prostate cancer. So what we're finding is that anoic signature as well as the DNA repair signatures are considerably anoic resistance rather, and DNA repair are considerably enhanced. And so the whole idea is that can these extracellular vesicles, can they give us some idea by profiling them by the RNA level about what is happening in the tumors they are derived from? And the answer is yes. Sometimes there is a differential expression, but sometimes they give us, so these can really provide a good foundation for liquid biopsies. And again, collaboration have great bioinformatics collaborators at the Mount Sinai, and this is Tina again. But this was published with work with Navneet as a collaborative effort, that these really from the castration-resistant prostate cancer cells, in addition to have something in common, they have their own unique RNA signatures that can be exploited or they can be identified as a markers of aggressive disease. Interestingly enough, and I know this is published, but I just want to draw your attention. What they do share, with the advanced prostate cancer cells, they do share the EMT reversal back to MET profile, as you can see here from the pathway association analysis. So sharing that and giving us some idea about what's happening in the EMT in the tumor by looking or studying the extracellular vesicles can really be profound in identifying the patients that are going to be respond to the combination treatments I talked about earlier. So hopefully what I managed to convince you today is some of our work and some of more recent work about how important is how the phenotypic dynamic in really impacting therapeutic vulnerability in these advanced prostate tumors. And this is happening this redifferentiation that is happening in a reciprocal way or manner or sequence, if you will, from EMT going back to MET, which is acquiring back the epithelial phenotype. This can actually enhance the therapeutic response in advanced prostate cancer, but it's all about the timing of paclitaxel given after androgen deprivation therapy, and then maybe paclitaxel, and then given the TGF-beta blockade, as well as kinase inhibition. So we are exploring that further now at the preclinical setting, but it's something that really can provide a new platform for treating patients with hormone-naive but advanced metastatic prostate cancer. And again, we learned a lot of lessons from the current clinical trials in terms of how we can stratify these patients, and maybe using liquid biopsies, we can really do that. And going back to the predictive signatures, about the gene changes associated not only with the EMT phenotype, but with anoikis, maybe the markers of the extracellular vesicle, which carry these anoikis signatures, and can really establish this treatment-induced or correlate with the treatment-induced changes within the microenvironment that are driving lethal prostate cancer. So gaining more mechanistic insights is extremely important in really understanding the treatment failure. And finally, the identifying new actionable targets or the interaction. We have a very active collaboration with Dr. Wei Yang, a proteomics expert who recently moved to Stony Brook from Cedars-Sinai. In terms of really identifying these interactions between BCL2 and kinases, I went through some of the precipitation data a little bit quickly here, but we are really something that we're exploring, that kinesins interacting with the transporter maybe, and being responsible for the taking carboxydoxal into the cells, and maybe failing of doing that in the absence of the transporter can be one of the mechanisms underlying the resistance to the drug. So the impact of trafficking is very important, not only the timing, but trafficking of the virus, and we believe that what kinesins are doing so by traveling or trafficking their cargo along microtubules. But also there are other probably intracellular transport proteins or partners that can be exploited further, and hopefully to find us with single cell RNA-seq. So I would like to, again, finish by acknowledging this team effort for the last several years. I'm very proud of my past trainees. I talked with some of you today about, and also people in the audience. Sarah Martin, who is now, one of the, actually, she's the global director for health policy oncology at Eli Lilly, and he's doing the pathologist at Memorial Sloan Kettering. Collaborators, my various chairman, and also the funding source, and various collaborators from MD Anderson to Stony Brook, as well as the Davis for the other things. And finally, this is the last slide. Again, this is Sarah Martin. She was an avid equestrian, competitive, she still is, dressage. I don't know how much time she has now. But she taught me a few things while she was working in the lab. Like says, when you do at the end of the presentation, at the end of the challenge, you go and present yourself to the judge. And really, we begin at the beginning and go until you come to the end, and then you stop. It's a pessimistic approach to really how we treat prostate cancer. Not really, but thinking about it, we begin at the beginning. We have to start thinking about, we treat these patients with hormone-sensitive prostate cancer, not when they are at the end, castration-resistant prostate cancer. And with that, I will thank you very much for your attention. Thank you for putting up with the heat in here. And yep. Okay. Yeah, elaborate. That's a very good question. And one of my colleagues is at BRC, as you know, from what I understand about BRC2 plays a major role in prostate cancer. BRC1 not only is, so it's something that we're exploring. This cell do express this, because again, this is done in the model. So BRC2 is expressed or mutated in this, but it's something that we're currently exploring. Olaparib has not been until recently, as you know, has been approved by the FDA for prostate cancer, but the clinical trials, I mean, it's done only for a very small number of patients. The first paper that came out by, I think, Pete Nelson. So now we're waiting for a larger, in terms of really correlating with the genotypic or the molecular makeup. So, but it's something that's why the trifecta is, I was very hesitant whether I should have used that last slide about the use of triple combination, the 3 drugs, because it's still very preliminary, but we're exploring that here. So it seems to be that the target, but it's really the timing of all 3 of them, and we're doing that in vivo work. So
I think there's some data actually where they
correct. Yes.
So I was wondering how to make that.
Yeah, there is some evidence. In fact, my lab was published a few years ago, a paper, that it was like exactly the opposite, that PARP loss led to, the loss of PARP led to high EMT and high progression of prostate cancer. This was published in Carcinogenesis. I couldn't get it published in any other. Well, actually, that's a pretty good journal, but I'm just saying the prostate cancer community didn't accept that, because at the time it was like, oh, PARP, the DNA repair is very much involved in prostate cancer. And this was we showed exactly the opposite. But lots of these, the PARP is involved in more aggressive disease. So in terms, and there has been, and for that, going back to your question about EMT, we show that loss of PARP is associated in that model with the induction of EMT, and therefore part of the resistance. So now we were, you know, again, we haven't looked at PARP in this model right now, but again, it goes back to the fact that, as we talked, that the EMT is such a dynamic phenomenon. So you need to capture that at the moment in time when it's really this epithelial phenotype, and then we hit that with this. Yes, but I'm familiar with Aruchi Mayan's data on that, but something that needs further exploration. I don't think it's totally clear about there is a connection between PARP or RAGA and EMT in other cancers too, not only in prostate. Yeah. Yes. Yeah, loss of eckatilin, yes. But you have to remember, I showed you that we want that to happen. No, we don't have increasing EMT. It makes the cells more aggressive. But it really is the time of. Are you asking whether there is an induction of EMT or?
Yeah, I guess. You might have some cell.
Correct. That can lead to the resistance. Yes. So it's something that is. That's why we need to do the studies in the in vivo component to see that you're right. You're looking with different subtypes, and again, these are homogeneous populations. But if, at the time, really, when we're looking at, for example, the triple combination after 24 hours, 48 hours of treatment, that loss of EMT can really promote that, and then maybe can promote all these drugs were given at the same time in the triple combination. So the timing is about really, and that's what we're doing right now in the animals. Yes, in vivo. But that's a very good point. Yes. Otherwise, you can inherently promote resistance. Yes. Yes.
Great talk. Anyway, you know, you're focused very on the cell components of it.
Yes.
But in the model system, seeing plasticity moving through functional acquisition of recipes, do you see alternatives like so, you know, when you break them apart, have you noticed any changes in integration?
So you are talking about characterizing the environment and that's what we're trying to do.
Cell, it might be now.
Yeah, that's what we're trying to do. That's why I drew that picture, because it's still very preliminary to a single cell. But as you know, it's very expensive, the single cell analysis. So we have a grant in that hopefully will be. But yes, it's extremely important to characterize the microenvironment. As you may know, prostate cancer, prostate tumors are very heterogeneous. That's right. And also they have. It's not just the microenvironment, not just endothelial cells, because we're focusing on endothelial cells. We're focusing on the immune cells, but also the stroma cells. So what I'm turning now, I don't know if anybody here is working on organoids, but in terms of really studying the microenvironment in a more, I guess, concise fashion, we're collaborating with, with people from Memorial, and maybe we're getting our own organoids. So the guy who was at, just moved to Cornell, was doing a lot of organoids, but we are, yes, it's very important, and that's what we're going to be doing, single cell sequencing, characterizing the AVs from the organoids, but also characterizing the various, and these are derived from tumors of various stages of treatment, and also in addition to the PDX, but this is the only way to answer that, yes. We're not ignoring yes, absolutely, that's an excellent question. We're not just focusing on one cell type, because you have to look at the microenvironment. You have to look at the dynamic.
Once it gets going,
yes. Yes. And one thing that I'm currently postdoc, I have Madison came from Australia, and her PhD was looking at macrophages in breast cancer. So now we're just trying to characterize. I know I talked with some of you about macrophages and how the role they can play. Yes. And again, even the EVs, I don't know how many of you believe in EVs or accept EVs as cellular vesicle, but we're finding some very interesting data about how these cells are, how these molecules or these small particles. Well, first of all, were identified by apoptotic bodies, because the apoptotic bodies are EVs. And when the cells undergo apoptosis, the first thing they secrete is this, apoptotic bodies that they have morphologically intact organisms, and then they're phagocytized either by macrophages or other. So That's a very good question. But I think the only way, and is the RNA-seq to identify these different subtypes. Everybody, that's a very good question. Thank you.
So I have a question.
Yes, please.
If I follow, it was a very fun and beautiful talk. Thank you very much.
Thank you.
And if I'm not mistaken, we saw examples of cells that were transitioning from epithelial to mesenchymal or mesenchymal to epithelial
Yes.
at the time when growth was suppressed.
Correct.
Which
Yes.
suggests that maybe there's an unstable epigenetic state that makes cells more vulnerable to certain therapies.
When they're in that particular state, yes.
Yeah. So have you
Yes.
looked at epigenetics, or do you think it's really intrinsically true that more differentiated cells are more vulnerable, or less differentiated cells are more vulnerable, or is it maybe just being in this transition that's the vulnerable state?
Yeah, I think more differentiated cells, well differentiated, acquiring the epithelial phenotype, are more vulnerable. I think that's a very good question. Actually, last year we wrote a commentary for Nature Communications about the epigenetic control of different subtypes in prostate. And as you know from Amina's work and other people, there is a lot of focus on the neuroendocrine phenotype, the emergence of neuroendocrine phenotype, and how different epigenetic mechanisms. I think there are certainly some epigenetic mechanisms we're currently just trying to define in terms of finding the appropriate model. I really think that overall the cells have to be in the well-differentiated state in order to become vulnerable to that. And EMT back to MET leads to that, but it's a very transient thing. So if you're asking whether there are cells that are already in that state and they can be targeted, probably yes. But we still need to, and there are some genes, not only for the neuroendocrine, as the pathologist, neuroendocrine phenotype or the emergence, there are a lot of in response to treatment. So, for example, when we give cabazitaxel alone, there is treatment-induced neuroendocrine differentiation. There is treatment-induced when you give that. So the timing is extremely important. That's why David Mulholland at Mount Sinai is using a lot of models of plasticity in terms of when to use it and when to really. I think before we move with all these combinations in the clinical trials, it's very important to have ways of monitoring, or that's why we turn to extracellular vesicles to capture those cells that are going to be in the state of one. But there are different subtypes. I don't think they're permanent. I think there is still even the differentiation stage. They're there, but they can be targeted. But I think those are they become more vulnerable. But definitely epigenetic phenomena, epigenetic changes can also. There are a series of genes that we can talk about that tomorrow. But yeah, definitely. Okay. Well, thank you very much, and